Hi, my name is Jessica Wang, and I'm a senior in biomedical engineering. I'm currently working in Dr. Meyer's biomembrane lab through the SURE program, and my research project this summer was to devise a method to enclose nanowells with spanning lipid bilayers. So planar lipid bilayers are important model systems that allow for the studying of membranes and membrane-associated proteins. In a cell, a lipid membrane contains many membrane-spanning proteins that play important roles such as signal transduction and transport of ions and small molecules in and out of the cells. So a model lipid bilayer that contains proteins of interest is thus a useful system for studying these membrane proteins. So a planar lipid bilayer is essentially a flat lipid bilayer that can be formed by different methods in different ways, for example, on a substrate or across a pore. So this image here shows a supported lipid bilayer on a solid substrate, which forms easily on clean hydrophilic surfaces like glass or silicon. So the larger goal of my project is to use spanning lipid bilayers, which would be a lipid bilayer placed over a pore or a nanowell in this case, to characterize a certain type of transmembrane protein called multi-drug resistance transporters. Now, MDR transporters are found in high numbers in cancer cells, and they're responsible for pumping out chemotherapy drugs out of cells, which makes the drugs less effective. So the idea is to be able to characterize these proteins with a system. Now, here we have a glass substrate with nanowells drilled into them, and the spanning lipid bilayer is going to be placed over them with these membrane proteins. So if the proteins can be positioned over the nanowells, we can fluorescently label substrates shown here, and as they pump the substrates into these wells, we can monitor the transport kinetics by measuring changes in their fluorescence. So using small nanowells lets us look at individual proteins instead of many at once. So that's the overall goal. This is an image of the nanowells that we're using. The nanowells are drilled into a cover slip by a laser, and each nanowell is about 200 nanometers in diameter and 3 microns deep. So this entire array is too small to be seen without a microscope. So the first step is to come up with a way to create the spanning lipid bilayers over the glass so that they span the wells. It seems simple, but it hasn't been. So it turns out that when I fuse these lipid vesicles onto the glass, instead of spanning the top of the wells, they actually follow the surface of the glass and tend to go into the wells, which isn't what we want. So this here is an image of the well array. You can see the small dots, which are the nanowells. And it's red because the lipids are labeled red and they're covering the glass surface. This is a cross-section of the cover slip on the right, and as you can see, the surface is covered by lipids and is red. And you can also see the wells showing up as red because the lipids have gone into the nanowells. So I've essentially spent my summer trying to figure out how to make the lipids span the nanowells instead of go into them. So one idea was to use electrostatic interactions by using charge repulsion. So glass is inherently negatively charged, and so if we can make the surface of the glass positively charged and leave the nanowells negatively charged by stamping polylysine onto the surface, then if we use negatively charged lipid vesicles, we can hopefully get the lipids to be attracted to the surface of the glass but repulsed from the nanowells. So that was the idea, and I've tried this using various amounts of charges and stuff shown in this image, but so far it hasn't worked. I'm currently trying to coat the inside of the nanowells with BSA, which is bovine serum albumin, which is a protein, and that's negatively charged, and it inhibits bilayer formation. But the difficulty lies in getting the BSA only in the nanowells and not on the surface of the glass, and the polylysine only on the surface of the glass and not in the nanowells, so that you have this charge difference. And so I'm still trying to figure that out. And another idea is to use gels as a physical barrier so that the lipid bilayers can't go into the wells at all because there's something there. The problem is that the wells are extremely small, and it's hard to know if the gel is actually forming and staying in the nanowells. So I've used agarose gels and polyacrylamide gels, which are water-based gels, which wouldn't inhibit any transport of substrate into the nanowells, but it would provide a physical barrier. I've tried using them in different concentrations and polymerizing them on the cover slip and then taking them off the surface in such a way that the gel remains in the wells but not on the surface. And so far, I really haven't gotten good results with this method either. So I'm just going to continue working in the lab and trying different variations of methods using charge repulsion and gels. And I'm also going to try different things such as using different methods of forming these bilayers, stamping and unstamping different surface modifications onto the glass, and just keep trying, and hopefully it'll eventually work. So thank you for your time.